When you look at the heart, you can ask two kinds of questions. The first kind is structural. Where are the chambers? Which valve lies between the left atrium and left ventricle? Where do the coronary arteries run? The second kind is functional. How does pressure develop? Why does a valve open and close? How does an electrical signal produce contraction? The first group belongs mainly to anatomy. The second belongs mainly to physiology. Clinical medicine becomes much easier when these two perspectives are connected. In this lesson, we will learn not only how anatomy and physiology differ, but also why neither subject is complete without the other. By the end of this lesson, you should be able to define anatomy and physiology in simple medical language, distinguish a structural question from a functional question, explain how biological structure supports function, and use this relationship in clinical reasoning. You should also be able to recognize whether a diagnostic test is mainly examining structure, function, or both. As you watch, keep one question in mind. If a structure changes, what function would you expect to change? And if a function fails, where would you look for the responsible structure or regulatory mechanism? Anatomy is the study of body structures and the relationships among them. It tells us what is present, where it is located, what shape it has, and what lies around it. Gross anatomy studies structures visible without a microscope. Regional anatomy examines all structures in one area, while systemic anatomy follows one organ system throughout the body. Microscopic anatomy includes tissues and cells. Anatomy is the map of medicine. It allows a surgeon to avoid a nerve, a radiologist to localize a lesion, and a clinician to predict which functions may be affected when a particular structure is injured. Physiology studies how living structures work. It focuses on function, mechanism, regulation, and response to change. Saying that the heart pumps blood is only the beginning. Physiology asks how electrical activity is generated, how contraction creates pressure, how pressure produces flow, and how the nervous and endocrine systems adjust cardiac output. The same principle applies to every organ. The kidneys do not merely make urine. They filter plasma, selectively reabsorb useful substances, secrete others, and regulate water, electrolytes, acid-base balance, and blood pressure. Physiology therefore explains the process behind the function. Use this comparison whenever the two terms feel confusing. Anatomy asks, what is it? Where is it? What is its shape, size, and relationship? Physiology asks, what does it do? How does it do it? What regulates it? With the heart, anatomy identifies chambers, valves, muscle layers, and vessels. Physiology explains filling, ejection, electrical conduction, pressure gradients, and cardiac output. These are different questions, but they belong to the same organ. In clinical practice, we move continuously between them. We identify the structure, understand its normal function, and then predict the consequence of disease. The structure-function principle means that physical form helps make biological activity possible. This principle appears at every level. Protein shape influences receptor binding. Cell shape influences movement or transport. Tissue arrangement determines strength, diffusion distance, or surface area. Organ architecture controls flow and coordination. On this slide, notice 4 examples: the thin alveolus for diffusion, the biconcave red blood cell for gas transport and flexibility, the intestinal villus for absorption, and the cardiac valve for one-way flow. Rather than memorizing the shape alone, always ask, what functional advantage does this structure provide? Alveoli demonstrate structure-function integration beautifully. Their walls are extremely thin, they provide a very large total surface area, and they are surrounded by pulmonary capillaries. These features reduce diffusion distance and create extensive contact between air and blood. Oxygen moves from alveolar gas into blood, while carbon dioxide moves in the opposite direction. If the barrier becomes thicker, as in pulmonary fibrosis, diffusion becomes more difficult. If surface area is lost, as in emphysema, gas exchange is impaired. The clinical problem is functional, but the explanation begins with altered structure. The small intestine and kidney show how structural specialization supports selective processing. Intestinal folds, villi, and microvilli greatly expand the surface available for absorption. Blood capillaries and lymphatic lacteals then carry absorbed nutrients through different pathways. In the kidney, the nephron is divided into segments with different epithelial features, transport proteins, and permeability. Filtration occurs at the renal corpuscle. Bulk reabsorption occurs in the proximal tubule. The loop of Henle helps create an osmotic gradient, and distal segments make regulated adjustments. The order and structure of these segments are essential to renal function. Structure affects function, but function can also reshape structure. Repeated exercise can produce skeletal muscle hypertrophy. Mechanical loading influences bone remodeling. Sustained pressure overload can thicken the ventricular wall. Repeated neural activity can strengthen or reorganize connections. These changes may initially be adaptive. For example, a thicker ventricle can generate more pressure against increased resistance. But compensation can have a cost. Hypertrophy may later impair relaxation and increase oxygen demand. When you see an anatomical change, ask whether it is the original lesion, a compensatory response, or a later consequence of prolonged physiological stress. Pathophysiology is the bridge between normal science and disease. Start with normal structure and normal function. Then identify what has changed. Perhaps a valve has narrowed, a receptor no longer responds, a nerve is compressed, or a feedback system is dysregulated. That disturbance changes function. Altered function then produces symptoms, signs, and test abnormalities. In aortic stenosis, structural narrowing of the valve increases resistance to outflow. The left ventricle generates higher pressure, hypertrophies, and may eventually fail to meet demand during exertion. The patient's symptoms become understandable when we follow the chain from structure to function to clinical effect. Diagnostic tests answer different questions. Structural tests show form, location, continuity, or tissue appearance. Examples include radiography, CT, MRI, endoscopy, and histology. Functional tests measure performance or regulation. Examples include ECG, spirometry, nerve conduction studies, and endocrine stimulation tests. Some tests combine both perspectives. Echocardiography can show chamber and valve anatomy while also assessing movement, flow, and pumping performance. A frequent exam mistake is choosing a test that does not answer the question. Before selecting an investigation, decide whether you need localization, morphology, performance, regulation, or a combination. Use this 5-step structure-function framework in clinical cases. First, locate the relevant structure. Second, state its normal function. Third, identify the mechanism that produces that function. Fourth, predict what happens if the structure is damaged or the mechanism is disturbed. Fifth, connect the prediction to the patient's symptoms, signs, and tests. For example, injury to a motor nerve affects a specific muscle movement. Anatomy localizes the nerve and muscle. Physiology explains how loss of motor signaling produces weakness. This framework reduces blind memorization because each clinical finding becomes a logical consequence. 3 exam traps are especially important. First, normal imaging does not always mean normal function. Electrical, biochemical, or regulatory disorders may occur before gross structural change is visible. Second, an anatomical abnormality may not yet cause symptoms because the body has reserve or compensation. Third, do not assume that one test provides every type of information. An ECG evaluates electrical activity, not coronary anatomy. A CT image may define structure but may not fully measure dynamic regulation. Read the question carefully and identify whether it is asking where the problem is, what the structure looks like, or how well it is working. Consider this case. A 72-year-old patient has exertional shortness of breath. Echocardiography shows thickened aortic valve leaflets, restricted opening, a high pressure gradient, and a thickened left ventricular wall. The structural lesion is the narrowed valve. The immediate functional consequence is increased resistance to ventricular outflow. The ventricle must generate higher pressure, leading to hypertrophy. Over time, filling may become impaired, and effective output during exertion may be inadequate. Anatomy identifies what is abnormal. Physiology explains why the patient becomes symptomatic. This is the structure-function method in action. Let us finish with 4 statements. Anatomy studies structure, location, and relationships. Physiology studies function, mechanism, and regulation. Structure and function are integrated. Specialized form supports specialized activity, while repeated functional demand can remodel structure. Pathophysiology explains how structural damage or disturbed regulation changes function and produces disease manifestations. In the next lesson, we will build the organizational map of the human body, from molecules and cells to tissues, organs, organ systems, and the whole organism. That map will help us understand how a change at one level can produce effects at every level above it.